The title of this talk is The Olfactory System. These are the objectives of this talk. You do not need to read them at this point. We will address each of them as we go along. This talk will be conducted in a question and answer format. The first question is: the olfactory nasal mucosa has pluripotential cells. A true, B false. This cartoon illustrates the nasal passage and some of its components. It is displayed here because in it you can see the area corresponding to the respiratory mucosa and the olfactory mucosa. This is an histological specimen of the olfactory mucosa. I will first concentrate in this area that I have now illustrated. The olfactory epithelium is classified as a pseudo-striated columnar epithelium. In the illustration, the pale cream colored cells drawn in the background forming a row are called sus tentacular cells. In the specimen, you can see them, with their dark nuclei forming a well-organized row. The sus tentacular cells are astrocytic lineage, and judging from their position and name, they support the other epithelial structures. In this illustration, in blue, you can see the olfactory sensory neurons, also called olfactory receptor neurons. In the specimen, they constitute the bulk of the cells below the row of sus tentacular cells. The dendrites of the olfactory receptor neurons end in a brush of immobile cilias. The cilias are emerged in mucus. In the illustration, the cells in pink are called microvillar cells. In the specimen, I have indicated to a cell that is likely to be a microvillar cell. In the illustration, the cream-colored oval structures are called basal cells. These cells are easily identified in the specimen because they form a line below the neurons and the nuclei have a more rectangular appearance. The basal cells are neuropluripotential stem cells. They have the lifelong capacity to generate olfactory receptor neurons, microvillar cells, and sus tentacular cells. In the illustration, the structure just indicated, looking like a chimney, corresponds to a Bowman's gland. You can get a glimpse of one in the specimen, as indicated by the arrow. Now, I'd like you to shift your attention to the lower segment of the specimen, which I have schematized and labeled lamina propria in this frame. I like to start by signaling the basal cells in the illustration and in the specimen. I have done this to keep our bearing in relation to the prior illustration. Below the row of basal cells in the illustration, in light and dark pink, we have the basal membrane. The basal membrane is hardly noticeable in the specimen. In the illustration, I am now indicating the olfactory ensheathing cells. They have been cut tangentially. In the specimen, they are cut transversely, hence they look very different. Olfactory ensheathing cells are responsible for the successful guidance of the olfactory axons of the newly formed olfactory sensory neurons throughout the life of an adult mammal. The blue lines in the illustration represent a collection of axons forming an olfactory nerve. The olfactory nerve can be clearly seen in this specimen. The axons with the olfactory ensheathing cells form the circular structure I am indicating now. The gray cells in the illustration corresponds to olfactory ectomesenchymal stem cell. I am now indicating one of them in the specimen. Olfactory ectomesenchymal stem cells have the same characteristics as the basal cells. They can generate neurons as well as other olfactory epithelial cells throughout adult life. Culture of basal and olfactory ectomesenchymal stem cells can hopefully one day be used to generate dopamine neurons and cure Parkinson, GABAergic neurons and cure Huntington, and acetylcholine neurons and cure Alzheimer's. So the answer to this question is A. Next question. Cortical input influences the sense of smell. A, true. B, false. This picture highlights the location of important olfactory structures. This is the olfactory bulb. These are the olfactory nerves going through the cribriform plate. This is a cartoonish representation of these structures. Cribriform plate, olfactory mucosa, and mucus. The mucus is the product of the secretion of epithelial cells and glands of Bowman. I have now added the body, dendrite, and immobile cilia of multiple olfactory neurons. And now I have removed the mucosa and the mucus so we can appreciate them better. I am now indicating to the bodies and dendrites of the olfactory neurons. The different color indicates the different and specific submodalities of a stimulant they perceive. I have now added the axons going through the cribriform plate. These axons, as they traverse the cribriform plate, form the olfactory filias, which join to form the true olfactory nerves. The axons of the olfactory neurons end in the olfactory glomerulus, where they make contact with dendrites of tufts and mitral cells. The glomeruli are represented in this frame as oval purple structures. At the glomerulus, multiple olfactory neurons axon conveying the same submodality of odor, here represented by the same color, here aqua, and here red, come together. It is important to stress that many olfactory sensory axons convey at a single mitral and tuft cell. It is also important to notice that although we have represented one glomerulus for each color, in reality there are many glomeruli subserving the same subset of odor modalities in the olfactory bulb. The mitral and tufted cells then send their axons through the olfactory tract. The mitral and tufted cells, in addition to being in contact with olfactory neurons, axons are in contact with granule cells. These neurons have the peculiarity of not having axons. All neurites are considered dendrites. It is important to recognize that the mitral, tufted, and granule cells receive input from the cerebral cortex and subcortical nuclei. These connections influence our perception of odors. These neurons also receive axon from tuft and mitral cells in the opposite olfactory bulb. These last connections are not illustrated in this frame. So the answer to this question is A. Next question. There is a different olfactory neuron for each odor. A true, B false. There seems to be 400 different primary odorants and a type of olfactory neuron for each of them. The existence of 400 primary odors seems extravagant if we take into account that there are only 3 primary colors and 5 primary tastants. The existence of 400 smell cells seems extraordinary if we take into account that there are only 4 types of retinal cells and 5 types of basic taste cells. Also amazing is that the number of different odors perceived by a human is over 10,000. This is easily conceptualized by thinking that 26-letter alphabet, when combined, can yield millions of words. I will further clarify this point using an imaginary example. One odor, let's say coffee, is the result of a stimulation of a set of number of olfactory receptor cells, each perceiving a subcomponent of the odor that, when simultaneously stimulated, are interpreted as the odor of coffee. The same sensory neurons with a single variation can, for example, detect the odor of bread, and some of the same neurons with a few additional ones may be stimulated by cigar odor. So the answer to this question is B. Next question. The axons of the olfactory receptor neurons are myelinated. A true, B false. I have now brought back the prior diagram of the neurons and glomeruli to highlight a single olfactory neuron, isolate it and enlarge it. I have now added a glia cell above the cribriform plate in the bulb and a vessel. I am now indicating to the axon of the olfactory receptor neuron. The axon is unmyelinated. Other structures in the olfactory receptor neurons are the axon hillock or trigger zone, the body, dendrite that ends up in a swelling called the olfactory vesicle or olfactory knob, and a brush of immobile cilia arising from the olfactory vesicle. So the answer to this question is B. Next question. Olfactory receptor neurons behave like fast adapting transducers. A true, B false. The olfactory neuron has the distinction of having the slowest conducting axons in the entire nervous system. They also have the distinction that they die in 1 to 2 months. But on the bright side, they are replenished by the transformation of basal cells and olfactory ectomesenchymal stem cells present in the olfactory mucosa. They also have a very beneficial quality: fast adaptability. When exposed to a bad smell for a prolonged period of time, as I am indicating in this frame, the receptor potential created by the receptor membrane of the olfactory neurons located in the immobile cilia will be short-lived, so the bad smell will not be perceived for long. So the answer to this question is A. Next question. The receptor potential is generated at the immobile cilia. A true, B false. To answer this question, I will refer back to the schematic representation of the neuron and single out three regions. The immobile cilia, where the receptive membrane is located, and the receptor potential is generated, represented inside the aqua rectangle. The trigger zone, which is at the beginning of the axon, represented in the yellow rectangle, and a segment of unmyelinated axon, represented inside the green rectangle. These three areas are fundamental for recognition of an adequate stimulus, transduction of a chemical energy to electrical energy, and transportation of electrical energy to the mitral and tufted cells, as we will see in details in the next few frames. So the answer to this question is A. Next question. Which of the following tasks takes place in the smell receptor cell? A. Recognition of a stimulus. B. Transduction of chemical energy to electrical energy. C. Generation of an electrotonic current. D. All of the above. Smell receptor neurons perform multiple tasks in order to achieve their primary function of adequately stimulating the second neurons in the olfactory pathway. The first task is recognition of an adequate stimulus. In a segment of the membrane of the immobile cilia, called the receptive membrane, we find the mechanism for stimulus recognition. In this frame, I added a figure with the components necessary for recognizing an appropriate chemical stimulus. The site of these elements is referred to as the odorant recognition site. At this site, we will find G protein coupled receptors embedded in the cell membrane. The interaction between a receptor and a specific odorant resembles the interaction of a key and a lock device. The receptor, the lock, only reacting to a specific basic odorant, the key. The second task consists of transduction of chemical energy to electrical energy. The receptive membrane is also involved in transforming the chemical stimulation into an electrical signal. This process is termed primary transduction. This process involves, in addition to the previously mentioned elements that I introduced when talking about the previous task, the ones I have added in this frame. This structure represents the membrane. The resting membrane potential is, let's say, as it is in most neurons, minus 70 millivolts. You can see the odorant represented as green dots. Now I am pointing to a G protein coupled receptor. The effector, second messenger complex, and now to a cation channel. The cation channel is a transmembrane protein that opens upon stimulation by the second messenger. Here I am pointing to sodium and calcium cations. Now that we have introduced all the elements in this illustration, we will explain their interaction with an experiment. The experiment starts by placing a needle intracellularly in the receptive area, as shown in this frame. The needle is attached to a grounded recording device that displays voltage, as represented here. At this stage, the membrane is at rest, so we will record the resting membrane potential minus 70 millivolts, as indicated here. When the appropriate odorant interacts with the G-protein coupled receptor, an intracellular element, the G-protein undergoes transformational changes that we will illustrate as coming apart from the receptor. This element then interacts with another element usually referred to as an effector. The effector then goes through configurational changes, leading to the activation of a second messenger. The second messenger then interacts with the cation channel and the channel opens. Then the cations present outside the cell go across the membrane, generating a receptor potential that may be small, as represented here, or large, as represented here. Whether large or small, the amplitude of the receptor potential is proportional to the intensity of the stimulus, as we will see in the next few frames. The introduction of a weak stimulus has three consequences. One is the opening of a small number of cation channels. The second one is the entry of a small number of positive charges into the cell, and the third one, the generation of a small receptor potential. I will now illustrate these consequences. This is the cell at rest. A small rise in membrane potential is reflected in this illustration by the increased membrane potential to minus 50 millivolts, and illustrated here. This is followed by the closure of the cation channels, the cessation of flow of cations, the activation of channels that expel positive charges, and the action of the sodium-potassium pump. As a result, the membrane will go back to the resting state of minus 70 millivolts, as illustrated here. The introduction of a strong stimulus will also have three consequences. One is the opening of a large number of cation channels. The second one is the entry of large number of positive charges into the cell. And the third one, the generation of a large receptor potential. I will now illustrate these events as I did for a weak stimulation. Again, we start with a resting membrane potential of about minus 70 millivolts. The opening of many cation channels brings about the entry of many cations into the cell, changing the membrane potential, in this case to minus 10 millivolts, as indicated here and illustrated here. Then the channels close. The action of other channels that expel positive charges from the cell and the action of the sodium-potassium pump brings the membrane potential to minus 70 millivolts, as illustrated here. The third task is the generation of an electrotonic current. I will again explain this task referring to a weak stimulus and a strong stimulus. The introduction of a weak stimulus, as is reflected here, generates an electrotonic current that fades in the distal dendrite, as indicated by the short arrow inside the dendrite. It is important to stress that the electrotonic current will not be recorded by the needle inserted in the distal microvilli. It will take needles inserted at multiple points in the path of the electrical current to record it. I will now illustrate the result of a strong stimulus. As you can see here, the resting membrane potential is minus 70 millivolts, in this case following a large receptor potential. I will remove the needle from the cilia to emphasize that electrotonic current is not recorded by it. I have now added the electrotonic current in the form of a white arrow. In case of a strong stimulus, the electrotonic current will advance in the distal dendrite with enough intensity as to move into the proximal dendrite, the neuronal body, and ultimately reach the axon hillock. The axon hillock, as you know, is the site in the neuron with the highest density of voltage-gated sodium channels. The fourth task is the generation in the region of the axon hillock initial axon segment of an action potential. The fifth task is conduction of the electrical energy from the initial segment of the axon to the distalmost region of the axon. And the sixth task is the release of neurotransmitters. So the answer to this question is D. Next question. An action potential generated at the axon hillock has the same configuration as an action potential generated further along the axon. A true, B false. An electrode introduced in the axon hillock in the case of a strong stimulus will record the initial action potential, as you can see here. Notice the hump, reflecting the gradual increase in membrane potential that occurs at the axon hillock as electrotonic current arrives from different areas of the soma at different times. This is so because although we have explained this phenomenon as the arrival of a single wave of electrotonic current, the product of a single stimulus, in reality, electrotonic current arrival to the axon hillock is not simultaneous for two reasons. One is that it usually takes more than one single event to generate a strong enough electrotonic current, and the second relates to the fact that electrotonic current has to travel through different pathways to reach the axon hillock. Once the first action potential is generated, the current created by the first action potential propagates electrotonically from one contiguous patch of axon to the next. Were we then to have placed a needle distally in the axon, as illustrated here, the advancing current will produce an action potential generated in the membrane patch under the electrode. As shown here, notice that this action potential does not have a hump. The lack of a hump reflects the simultaneous arrival of electrotonic current to the patch of membrane and its simultaneous depolarization. So how do we put these effects together? A strong stimulus raises the membrane potential to minus 10 millivolts. The channels close and the receptor potential dies down. Yet the receptor potential has generated an electrotonic force or current sufficient to reach the axon hillock and generate the initial action potential with a hump. The current produced by this action potential is then transmitted electrotonically from one contiguous axon segment to the next, bringing the distal segment to threshold, thus triggering an advancing action potential. The action potential captured by the needle does not have a hump, as you can see here. So the answer to this question is B. Next question. All olfactory receptive neurons use cyclic AMP for transduction. A true, B false. Olfactory sensory neurons have two more of primary transduction processes. The first one and most common one is shown in this frame. I will now enlarge the figure I have just introduced and introduce the legend for each component. As you can see here, the arrow points to the odorant receptor binding being stimulated. This brings about a confrontational change in the guanine nucleotide binding protein expressed in the olfactory sensory neurons, and subsequent activation of the calcium-dependent adenyl cyclase, which brings about the conversion of ATP to cyclic AMP, which interacts with cyclic nucleotide-gated receptors and triggers the opening of the cyclic nucleotide-gated cation channels and entry of sodium and calcium. The increase in intracellular calcium then triggers the opening of chloride channels, and chloride and negative ions leave the cell, thus furthering rising the membrane potential. The second transduction process mechanism is presented in this frame. It uses guanylyl cyclase. In this frame, I have enlarged the diagram and include the legend. As you can see in this frame, the arrival of an appropriate stimulus to the guanylyl cyclase type D receptor produces a confrontational change that activates guanylyl cyclase. Activated guanylyl cyclase transform guanosine triphosphate into cyclic guanosine monophosphate, which triggers the opening of special cyclic nucleotide-gated channels and the entry of sodium and calcium. So the answer to this question is B. Next question. The olfactory tracts are constituted only by the axons of neurons in the ipsilateral olfactory bulb. A true, B false. This is a photograph of the orbitofrontal regions of the brain. Indicated by the magenta arrows are the bulbs, which are part of the paleocortex. This is an H&E stained histological specimen of the bulb. In it, you can see the olfactory nerve layer, the glomerular layer, the external plexiform layer, the internal plexiform layer, and the granular layer. The thin black arrows indicate the olfactory tracts. I have now introduced a transverse cut of the olfactory tract from another specimen. In it, you can see a triangular structure representing the olfactory tract itself and adjacent cerebral cortex. In the tracts, you can see axons originated in neurons in the ipsilateral and contralateral bulbs, as well as axons from cortical and nuclear cerebral structures. In addition to that, you can also see neurons. These neurons constitute the anterior olfactory nucleus. So the answer to this question is B. Next question. Electrical signals traveling the olfactory pathway reach the cortex prior to reaching the thalamus. A true, B false. This structure represents the olfactory bulb and the olfactory epithelium. The ellipse just introduced represents the lateral olfactory tract. At the olfactory bulb, I will represent tough cells that, as you can see, travel in the olfactory tract, reaching and contacting with anterior olfactory nucleus. The neurons in this nucleus will send axons to the contralateral olfactory bulb. The olfactory tubercle is located immediately caudal to the retrobulbar region of the anterior perforated substance. It is a relay station for olfactory information from the olfactory bulb and distributes this information to adjacent neurological areas. Tuft cells axons will also reach the olfactory tubercle. From the tubercle, fibers will go to the thalamus, and from the thalamus to the orbital frontal cortex. Fibers from the olfactory tubercula will also reach the prefrontal cortex. In the bulb, I will also represent mitral cells. Mitral cells axons will also send fibers to the anterior olfactory nucleus and the olfactory tubercles, but also to the piriform cortex. Neurons in the piriform cortex send axons to the thalamus and the prefrontal cortex. Fibers from mitral cells will also reach the amygdala. Axons of the neurons in the amygdala contact by mitral cells, then send their axons to the thalamus, prefrontal cortex, and hippocampus. Mitral axons also reach the entorhinal cortex. Entorhinal neurons contact by mitral cells send axons to the thalamus, prefrontal cortex, and hippocampus. In addition, the olfactory bulb also houses the accessory olfactory bulb. This area of the olfactory bulb receives axon from the vomeronasal organ. Mitral cells in this area make contact with the amygdala. The function of the vomeronasal organ is probably related to mate choosing for procreation. So the answer to this question is A. Next question. Which of the following is not true regarding medial olfactory stria? A. Responsible for autonomic responses. B. Send fibers through the anterior commissure to the contralateral olfactory bulb. C. Connects with septal nucleus. D. Some of its fibers reach the thalamus. The medial olfactory stria is responsible for autonomic responses associated with smell, such as an increase in salivation and gastric peristalsis, and secretion in response to smell of food or vomiting when presented with foul smell. Axons from mitral and tufted cells traveling in the medial stria either cross to the contralateral side by way of the anterior commissure to reach the contralateral olfactory bulb, or end up at the ipsilateral septal nucleus and paraterminal gyrus. Neurons contacted by the olfactory fibers in these regions travel in the olfactory hypothalamic tegmental bundle to reach the dorsal vagal nucleus. This circuit is concerned with the peristalsis and gastric secretion. Or they go via the medullary stria to the habenula, where they make contact with neurons that send axon to the superior and inferior salivary nucleus. So the answer to this question is D. Next question. Which of the following structures is in the frontal lobe? A. Entorhinal cortex. B. Piriform cortex. C. Olfactory tubercle. D. Amygdala. This photo corresponds to the inferior view of the brain. I will now enlarge the area inside the rectangle, as you can see here. I have now listed some of the structures you can visualize: the olfactory bulb, the olfactory tract, the usual location of the anterior olfactory nucleus, the medial olfactory stria, the lateral olfactory stria, The arrow points to the area of the olfactory tubercle. The tubercle is prominent in lower animals and usually not perceptible in most humans. The arrow is now indicating to the piriform cortex, called so because it looks to some to have the shape of a pear. In human anatomy, the piriform cortex consists of the cortical amygdala, uncus, and anterior parahippocampus gyrus. Another term for the piriform cortex is the piriform lobe. And now I am pointing to the entorhinal cortex. It is important to stress that the term piriform cortex and entorhinal cortex are used differently by different authors and sometimes differently even by the same author. And the last labeled structure is the amygdala. Its deep position is represented by the magenta circle. So the answer to this question is C. Next question. The percentage of olfactory cortex is less in dash A. Sheep, B. Adolescence, C. Adult, D. 13-week fetus. In this lateral view of a sheep brain, the magenta area corresponds to the rhinencephalum. In this lateral view of the human cerebrum, we do not see it. These are inferior views of a sheep brain, a 13-week-old human fetal brain, and an adult brain. As you can see, percentage-wise, the human brain has the smallest area occupied by the rhinencephalon. This is a drawing of the inferior surface of a human brain, highlighting the main divisions of the rhinencephalus at this age, which is 13 weeks. Notice the presence of the piriform lobe, which encompasses the gyrus ambiens and the gyrus semilunaris, as well as the giriform diagonal gyrus. This gyrus ends up becoming the horizontal component of the diagonal band of Broca. So the answer to this question is C. Next question. Which of the following is more common in patients with transient epileptic amnesia? A. Dysomnias, B. Olfactory agnosia, C. Phantosmias, D. Hyposmia. Let's talk about definitions. Dysomnias means distortions or pathologic alterations in oral perception. We can think of dysomnias as oral illusions. Olfactory agnosia implies an inability to distinguish odorants apart from one another. Everything smells the same. Anosmia or hyposmia means an inability or diminished ability to smell. Phantosmias implies an spontaneous experience of a smell in the absence of an odorant, or odorant hallucinations. Phantosmias occur often in patients with transient epileptic amnesia. Transient epileptic amnesia is characterized by recurring amnestic seizures in which memory functioning is disrupted over a brief period of time while other cognitive functions remain predominantly intact. So the answer to this question is C. Next question. Phantosmia is more often present in patients with dash A. Alzheimer's, B. Parkinson's disease, C. Schizophrenia, D. Meningiomas of the olfactory groove. Anosmia is associated with Alzheimer's disease, Parkinson's disease, and olfactory groove meningiomas. Patients with schizophrenia may present with anosmia but also with phantosmia. So the answer to this question is C. Next question. Which of the following conditions results from improper development of the nasal plaque and or migration of gonadotropin releasing hormone neurons? A. Alzheimer's disease, B. Parkinson's disease, C. Huntington disease, D. Kallmann syndrome. Kallmann syndrome is a rare genetic condition often mentioned in exams. The condition is characterized by hypogonadotrophic hypogonadism, ananosmia, or hyposmia. Kallmann syndrome is due to failure of the nasal plaque to generate olfactory cells in gonadotropin releasing hormone neurons, or it may be due to failure of gonadotropin releasing hormones to reach their normal destination. The MRI of patients with Kallmann syndrome often lack olfactory bulbs and tracts, as you can see in this frame. Here, the red arrow indicates the absence of, whereas the green arrow, the presence of olfactory tracts. So the answer to this question is D. Thank you very much for your attention.